Hi, my name is Beth Taylor, and the topic of the class today is DNA and genealogy, the basics, where this is just kind of an introduction and overview about DNA and how it is used by genealogists. So the first question we want to ask is, what is DNA itself? DNA is an acronym that stands for deoxyribonucleic acid. I promise there's no quiz, you don't have to remember that, but just to know that DNA actually has like I said, has a longer word. DNA is the genetic code that defines your biological characteristics. So it's kind of like that foundational, almost like a computer code that tells your body how to run. A DNA is made up of strings of what are called bases, or sometimes it's called nucleotides, that are paired with their counterparts. Nuclear or DNA is found in kind of two places, or at least the DNA that we talk about for genealogy purposes. First, you have nuclear DNA. Nuclear DNA is found within the nucleus of the individual cells within your body. The nuclear DNA that is composed of those base pairs or nucleotides, which are twisted into the kind of double helix pattern, and then they're coiled into packages called chromosomes. And humans have 23 paired chromosomes, so a total of 46 chromosomes. And of those 23 paired chromosomes, 22 of those pairs are numbered autosomes, and the final pair of chromosomes are the sex chromosomes, which defines whether you are biologically male or biologically female. So here is an example of what DNA looks like under an electron microscope. So you'll notice that they're just numbered, starting with number 1, 2, 3, and so forth. And chromosome number 1 is just the largest, the longest. That's why it got the first number. The other type of DNA that we talk about is mitochondrial DNA. Mitochondrial DNA is found in the mitochondria in your cells. Mitochondria are the cell's powerhouses where sugar is converted into energy. And mitochondria are located outside of the nucleus of the cell, and there are actually multiple copies of mitochondria within each cell. Mitochondrial DNA, interestingly enough, actually have their own DNA. Okay, so the next thing that we want to discuss is, why is DNA valuable for a genealogist? Why do we find it valuable, and why are we all talking about DNA? Well, I like to think that there's kind of two things to consider when you want to think about why DNA is valuable. First off is that DNA is actually inherited. So I love this idea that here I am spending a lot of time trying to understand my ancestors and learn who they were, and yet I have something that's part of me that I inherited from my ancestors. I think that's just an awesome idea. But the way that DNA is inherited is you get half of your chromosomes from mom and half your chromosomes from dad. So when we say they're paired, we literally mean one set from mom, one set from dad. So from dad, you do get one set of autosomes, so 22 autosomes. And then if you are biologically female, you receive an X chromosome from your father. If you're biologically male, you receive a Y chromosome from your father. Now from mom, again, you get 22 autosomes, and everyone receives an X chromosome from their mother. And then mitochondrial DNA is always inherited from mom as well. Everyone has mitochondrial DNA, but it's always inherited along the maternal line. Now, DNA by itself is not that valuable. Yes, you can look at your DNA, but you can't really figure anything out about it unless it's compared with other people. So really the value in DNA comes when there is another person who has also taken a DNA test, and your DNA and their DNA is compared. So the basic idea behind this is that if two people share DNA, so they share segments of DNA that are in common where the nucleotides are identical, or, you know, with looking at the Y chromosome, there's not a lot of variations between, you know, the two chromosomes, then the idea is that there is a relationship between those two people. And based on the amount of shared DNA or how closely the DNA matches, a relationship can be predicted. However, all the DNA is indicating is that there is a relationship, and that the relationship is probably, you know, this kind of a relationship. However, DNA cannot tell you exactly what that relationship is. So that's when we turn to traditional paper trail research to compare family trees and see if you can spot the common ancestor, or what we call the MRCA, or most recent common ancestor. That is, and that's a person that appears on both persons' trees, both DNA testers' trees. So there are 4 types of DNA used in genealogy. Let's take a look at each of those in a little more detail. First, we have Y-DNA. And when you're looking at your fan chart, this is the line of people that Y-DNA relates to. And remember that the Y chromosome is only carried by men. And because it's only carried by men, it has to be inherited from your father. If you have a Y chromosome, it had to come from dad. So that Y chromosome is inherited from your father, who inherited it from his father, who inherited it from his father, and so forth. So if you compare the Y chromosome of 2 men, you should be able to determine whether they have a common patrilineal ancestor or an ancestor along the paternal line. If the closer the chromosomes match, then the DNA company returns something called a genetic distance, and the smaller the genetic distance, the closer the ancestor is. The larger genetic distance, that common ancestor is probably further back in time. And when the Y chromosome is tested, what happens is they actually test specific locations on the Y, and these are called markers. So when you order a Y-DNA test, you'll order a certain number of locations to be tested: 37, 67, 111. The next type of DNA we want to talk about is mitochondrial DNA, or mtDNA. mtDNA relates to this line along on your fan chart or pedigree chart. Now, mitochondrial DNA is carried by everyone. Everyone has to have mitochondria. But that mitochondrial DNA is inherited from your mother, who inherited it from her mother, who inherited it from her mother, who inherited it from her mother, and so forth. So again, the theory, like with the Y, is that if you compare the mitochondrial DNA of 2 people, you can determine if they have a common matrilineal ancestor or an ancestor along the maternal line. One thing to note about mitochondrial DNA, however, is that it mutates slower than other types of DNA. What this means is that 2 people can have identical mitochondrial DNA, but the common ancestor may be further back in time. When mitochondrial DNA is tested, there are 3 different regions tested, what are called HVR1, HVR2, and the coding region. The testing offers usually will test either HVR1 and HVR2, or test all 3 regions. And of course, if you test all 3 regions, the better your results, but that does increase the cost of the test. Next, let's talk about autosomal DNA, or ATDNA. Autosomal DNA is inherited from most of your ancestors. So this isn't a true representation of which of your ancestors you got autosomal DNA from. This is just kind of a possibility. So autosomal DNA consists of the 22 numbered autosomes that we talked about earlier. And of those 22 pairs of autosomes, you got 1 set from mom and 1 set from dad. Now, before you got that DNA from mom and dad, you don't get complete chromosomes from, mom's parents. Instead, what happens is that the chromosomes kind of line up and exchange data before they split and 1 chromosome gets passed on to you. What this means is that that you don't get like an equal percentage of DNA from each grandparent. It's kind of more random. So you'll get some percentage from each grandparent, but that could be a little bit skewed. So this process of that exchanging data is called recombination. So because the chromosomes are recombined before you inherit them, it means that you kind of get a more random percentage of DNA from each of your grandparents and great-grandparents and so forth. Autosomal DNA can be used for about the last 5 to 7, maybe up to 9 generations. But beyond that, it's a lot harder to use for genealogy purposes. Now, the law of because of statistics, because you're only getting half of the DNA at each generation, you cannot actually inherit DNA from all of your ancestors. And I'm talking, all of your ancestors going back hundreds and hundreds of years. So some of your ancestors do drop off your genetic tree. And because of this, you're actually only guaranteed to share DNA with a second cousin or closer. So it is possible to have a known third cousin and you have a paper trail connection, but you don't share any DNA. That's okay. Autosomal DNA is measured in centimorgans. It's just a unit of measurement. The more centimorgans you share in common with somebody, the closer the relationship. The last type of DNA we want to talk about is xDNA. As we mentioned earlier, xDNA is inherited it has a unique inheritance pattern. If you have an X chromosome, actually everyone has an X chromosome from mom, that X was recombined from both of her parents. So that X can have DNA from both grandma and grandpa. However, if you received an X chromosome from dad, and you only receive an X chromosome from dad if you're biologically female, that X chromosome cannot come from your grandfather, it has to come from your paternal grandmother. So if you're biologically female, you have an X chromosome from your paternal grandmother that's kind of been inherited intact. And this is all because xDNA cannot be passed from father to son. Because Y DNA is always passed from father to son, xDNA cannot be passed from father to son. So the theory is that, or the idea is that if you share matching segments on the X chromosome, your common ancestor is actually going to be along specific lines. So because of this, you only can share xDNA with certain ancestors. You can't share it with all of your ancestors. So if you're biologically female, this is your xDNA fan chart. And if you're biologically male, this is your xDNA fan chart, meaning these are the ancestors with whom you could share xDNA. Okay, so there are the 5 companies that we talk about for genealogy purposes all offer a lot of different feature sets, but there are 2 main things that they offer, and I just want to focus on each of those for a moment. First off, all of these companies are going to offer some kind of an ethnicity or admixture results. Sometimes people refer to this as your percentages. It says I'm 27% German and 31% French and 18% Scottish, or whatever the company is using. So these ethnicity percentages, like I said, are offered by all of the DNA companies that we kind of use for genealogy. The way these percentages are determined is based on reference samples. So the DNA company goes out and compiles or gathers DNA samples from specific regions around the world, and they create something called a reference sample, and then they compare your DNA to those reference samples. And based on how similar your DNA is to those reference samples, along with some complex algorithms, they're going to then give you your percentages. One thing to note is that DNA companies actually all use completely different reference samples, which means that they are all going to give you different results, and they are regularly being updated, meaning you're regularly going to get, or you'll get changes. So it's not that your DNA sample changed in any way at all. It's just that they're refining and tweaking their algorithms, or they're adding new reference samples, trying to get those percentages to be more accurate, is the idea. Now, the other kind of result that DNA companies offer or will return are match lists. And this is where I think there's the most value for a genealogist is in using your match lists. So a match list is just a list of people with whom you share DNA. And the people on the list are determined differently whether you're looking at an autosomal DNA match list or a Y or mitochondrial DNA match list. So on an autosomal DNA match list, people are listed based on the amount of shared DNA, the amount of DNA that they share in common. Remember that autosomal DNA is measured in centimorgans, and the more centimorgans you share in common, the closer the relationship. Y DNA and mitochondrial DNA, those two types of DNA are, the relationship is based on how closely the DNA matches, the number of differences between two DNA samples. So the fewer the differences, and those differences are listed as a genetic distance. So the lower the genetic distance, then the closer the relationship when you're working with Y DNA and mitochondrial DNA match lists. Now, based on that shared DNA, the DNA company may predict some kind of a relationship. Again, the theory being that if you share DNA, we're assuming a relationship exists. So at that point, it's up to you to determine what that relationship is. The DNA company may provide some tools that give you some suggestions on what that relationship is, but it's really up to you to evaluate those suggestions or to go explore family trees and to identify the relationship. Now, you can do this in several different ways. The easiest way is if you know your family tree, and the match that you're looking at has a family tree that you can look at. It's pretty easy to go through, compare the trees, and find the common ancestor. Now, it's also possible that your match has the beginnings of a family tree, but they haven't filled it out. In that case, you may have to spend some time researching their family tree to try and figure out where the common ancestor is. You may need to communicate with your matches to kind of collaborate with them and see if they'll provide you enough information that you can, again, find the common ancestor, or if all else fails, you can at least use the shared matches or in common with features to determine what other matches on your match list share DNA with you and this match, and at least suggest which family line that your match is related to. So at least you can mark and say, well, this match is on a family line I'm not researching right now, so I'm not worried as much about how we're related. Now, once you start determining relationships, you want to be able to describe those relationships. And relationships are traditionally described using the term cousins, first cousin, second cousin, third cousin. However, it's also important to know when a cousin is a removed cousin, a cousin removed, or a half cousin. So let's take a quick look at how you can determine which cousins are removed and which cousins are half cousins. So here's kind of an example family where we have Martha, who married first John and then second Frederick. And by John, she had children named William and Grace. By Frederick, she had a daughter named Catherine. And William had a daughter, Elizabeth. Grace had a son, Joseph. Catherine had a daughter, Anna, and so forth. So how do we figure out the relationships between the people on this chart? Let's start by looking at Thomas and Arthur. So Thomas and Arthur share two ancestors in common, meaning they share John in common and they share Martha in common. So that means Thomas and Arthur are full cousins because they have two ancestors in common. From there, we also want to note if they're in the same generation, meaning Thomas and Arthur are each 3 generations if you count down to William to Elizabeth to Thomas, Grace to Joseph to Arthur. They're 3 generations down from John and Martha, meaning they're at the same generational level, meaning that they are just full cousins. In this case, they're second cousins. Now, how about if you're looking at 2 people who don't have 2 ancestors in common? So in this case, let's look at Margaret and Alexander. So Margaret and Alexander only have 1 ancestor in common, meaning they only have Martha in common, since they have Martha had married twice, so they descend from different husbands. this means that Margaret and Alexander are half cousins, not full cousins. However, Margaret and Alexander, again, are in the same generational level, so we refer to them just simply as half cousins. In this case, they are third half cousins. Now, what happens when two individuals are in different generations, meaning that they're a different number of generations down from the common ancestor? For example, Thomas and Margaret. So Thomas and Margaret definitely have two ancestors in common. They're full cousins. However, because they're not in the same generational level, we can't just refer to them as either second cousins or third cousins. We actually talk about them as cousins removed. And what all this means is, by removed, is that they're removed a generation from the common generational level. So in this case, we're going to go to the closest common generational level, which is Thomas and Arthur. So we go up the generational levels, not down, when determining a relationship like this. So we say that Thomas and Arthur are second cousins, but Margaret is one generation removed, or one generation down, from Thomas and Arthur, meaning Thomas and Margaret are second cousins once removed. So we look at that second cousins being Thomas and Arthur, and then Margaret's one generation removed from that. Second cousins once removed. Does that all make sense? Well, try it on your own. So stop for a second, consider how are Elizabeth and Alexander related. So Elizabeth and Alexander only share one ancestor in common, Martha, so they are going to be half cousins. And Elizabeth and Alexander are different generational levels. So we're going to go up 2 generations to Anna, and notice that Elizabeth and Anna would be half first cousins. And then we count down the number of generations that Alexander is, so he's 2 generations down. So Elizabeth and Alexander are first half cousins twice removed. Okay, so the last thing we just want to mention are the testing companies. There are 5 big testing companies that we refer to for genealogy purposes. And the reason that we use these testing companies is because they offer both ethnicity estimates, and they also offer a match list. Those 5 companies are FamilyTree DNA. FamilyTree DNA does an autosomal DNA test, as well as mitochondrial DNA and Y-DNA tests, and offer match lists for both mitochondrial and Y. They are the only company to offer match lists for mitochondrial and Y. There's 23andMe, who also offers a health feature, or you can get health data from 23andMe. There's Ancestry DNA, MyHeritage DNA, and Living DNA. And all 3 of those companies primarily offer autosomal DNA testing. 23andMe and Living DNA will also provide a haplogroup. We didn't really talk about haplogroups, but it's kind of your deep ancestry based on the Y and the mitochondrial, but they do not offer match lists. So those are kind of the 5 big DNA testing companies. You're going to want to investigate all of them and what they have to offer before you make a decision which testing company you want to start with. You'll find a lot of genealogists will end up testing with all 5 companies. So that's our class for today. Hope you enjoyed it. Hope you learned a little bit more about DNA and how you can use it for genealogy. Hope you have a wonderful day, and thanks for participating in RootsTech Connect.